Hi everyone. In today's session, we're going to be talking about the male reproductive system, and then we'll talk about the sexual response in males and females, as well as the genitalia, internal and external genitalia development towards the end. So let's go ahead and look at the male reproductive system first. Testes are the primary reproductive organs for males. And then the accessory organs that are important in male reproductive system are ducts, and there'll be several different names for different ducts. So that will be an important thing to remember, which duct is which. There are going to be several different tubules, accessory glands, as well as penis is also an accessory structure in the male reproductive system. So testes are the male gonads. They are located in scrotum, and scrotum provides cooler environment for the testes, which is necessary for the sperm development and maturation. The scrotum is composed of external layer of skin, a thin layer of superficial fascia, and dartos muscle. And this muscle helps with temperature regulation. There's also a cremaster muscle, and we'll see that actually on the second image that I will have after this one. So before I go into the spermatocord, I want to talk about this image just a little bit. This is the sagittal section of the male reproductive system. And what you can see here are the testes right here. So you can see the scrotum surrounding the testes, and they are not internal organs like the ovaries were in females, which are located in the pelvic cavity. Testes are outside of the body, and this is important again for that temperature regulation, because the proper temperature regulation for sperm formation is slightly lower than the internal body temperature. We can see the penis on here. We see some glands here that we'll be talking about. So this is, on this view, this is your prostate gland, your bulbourethral gland, that's this one right here. That's a paired gland, actually, it's a pair of them, but we only see one from this view. And then a seminal vesicle, which is this kind of wrinkly gland right here. And then you see this long tubule right here. This is your vas deferens, also known as the ductus deferens. And then this wider area of that vas deferens is known as the ampulla of the ductus deferens, or the vas deferens. Okay, so that should be pretty straightforward as far as this image goes. So let's go ahead and switch to this view here. And here we can see the spermatocord really nicely. So spermatocord is a structure that houses the vas deferens. It houses vessels that are going in and out of the testes, as well as some nerves. And those muscles that I was mentioning, we have the cremaster muscle, which is this one right here. And then the dartos muscle is a flat, kind of like a sheet-like muscle in the scrotum. So that's this muscle right here. Both of these muscles would contract in a colder environment. So when it's cold outside, these muscles would contract and bring the testes closer to the body. And then when in the during warm conditions, then these muscles relax and they allow for testes to be a little farther away from the body. So it's a temperature regulation right there. There are some other layers here that are involved in the scrotum, as well as the spermatocord. They're not super important there, but what is important here and that I want you to know is on this left side right here, notice the blood flow right here in that spermatocord. The blood flow and the way that the blood vessels are arranged within the spermatocord is another way how males regulate the temperature of the testes. So it's a little difficult to see for the artery, but there's just a single artery that's bringing blood to the testes. It's just one vessel, single artery. But then you can see that the veins there's not just a single vein running along it. The veins are actually arranged in a venous plexus, or known as the pampiniform plexus here. And this plexus surrounds that artery, that single artery. So it's kind of like a meshwork of veins surrounding that single artery in the center. And The importance of this is that the veins carry blood that has been cooled down already. It has that cooler temperature of the testes. The artery that's coming from the body into the testes, it's bringing warm blood. And so as the blood is passing into the testes and it's surrounded by this cooler blood, the blood gets cooled down before it reaches testes. It's kind of like a like air conditioning, but it's not air, right? And so it cools down that blood. The colder blood that's leaving the testes cools down the warm blood that's coming in through that single artery. Okay. What else do we see here? Now we can see the epididymis here. That's this structure right here. It's kind of like a comma shape, structure right here. And we'll talk about that here in a moment. Okay. So that's spermatocord. So let's look closer at and this is what the spermatocord would look like in the cadaver. You see the spermatocord in one piece right here, and then this is a dissected spermatocord, so you can see the structures within it. Okay. This image here, we're going to basically start talking about the pathway of sperm through the male's body. So where do the sperm originate and how do they move until they leave the body here? So this image shows us the testes, and each testes is covered by the visceral and parietal layer of tunica vaginalis. And the tunica vaginalis is this. You can see it's folded. It's basically a single membrane, but it's again folded onto itself. And so you have that visceral and parietal layer there. And then underneath that, you have tunica albuginea, and that's this layer right here, this layer, or you can see that, right here is that cross section of that layer. And that's tunica albuginea. Tunica albuginea is a fibrous capsule. It covers each testes. It thickens in the posterior margin, and it projects inwards into the organ. And that region is called mediastinum testes. That's this area right here. And mediastinum testes is going to house reed testes, okay? The tunica albuginea also forms connective tissue septa, and these are going to subdivide the testes into lobules. So you can see that the tunica albuginea has projections. Let me pick a different color here so that it's obvious, okay? So these projections right here, those are septa. And septa basically create little compartments inside of the testes. And these compartments are known as lobules. Each lobule contains up to 4 seminiferous tubules, okay? So lobules have up to 4 seminiferous tubules, and seminiferous tubules are the origins of sperm, okay? Seminiferous tubules is where the sperm are formed. And what I mean by this is going to be specifically within the wall of the tubule. So there are 2 types of cells that form the seminiferous tubule. They are Sertoli cells, and Sertoli cells are your supportive cells. They provide nourishment and also release inhibin whenever needed. And there are tight junctions between these Sertoli cells that create a blood-testes barrier. Blood-testes barrier is important because when you think about the structure of the sperm, sperm is in the end going to be a cell that has half of the genetic material. It doesn't look like the other body cells. And so if immune system cells saw sperm, they would know that this is a messed up cell. This is not looking normal like your other cells. And so blood-testes barrier is important for the immune, so that immune system doesn't attack your sperm. Okay. You can see, like, if we're looking at the cross section of the seminiferous tubule, here it is. Okay, so this is the border of one tubule, so we don't see the whole tubule there, but most of it. And notice the thickness of the tubule. Okay, this to this, and then this in the yellow, that's the lumen of the tubule. Okay. So the seminiferous tubules are are rather thick. The Sertoli cells are large, and we actually notice that you can see these cells that are towards the outer part of the tubule are tend to be larger, and then they get smaller and smaller as we move this way. Okay. And then ultimately they become sperm. And these little black dots here that you see, I'm going to do them in orange. Those are heads of the sperm. Okay. And then all these lines that you see here, those are the tails of the sperm. Okay. And that's going to make sense. Let's look at just to kind of get an idea in a diagram version here. The Sertoli cells are these cells. Okay, see, it's a huge cell compared to the sperm. Okay, all of this is the Sertoli cell. Okay, this purple one. And then the other type of cells that we see in the seminiferous tubules are the germ cells or the dividing cells. Okay, when we say germ cells, we don't mean, you know, like germs that get on your hands and stuff. We mean the dividing cells here. And so those are the cells that are becoming sperm ultimately, okay? So two types of cells that you find within the seminiferous tubules are Sertoli cells and the dividing cells, the germ cells. There is another type of cell that is found in the testes, not in the tubule, but in the testes. And those are cells that are found between the seminiferous tubules. These are known as the interstitial cells or your Leydig cells. Leydig cells are the cells that produce testosterone. These are here, okay? And these are outside of the tubule. They're not part of the tubule, okay? So going back to the testes here, because we're going to be talking about some other tubules while we're still here. So we mentioned that each lobule has up to 4 seminiferous tubules. The sperm, once it's produced in that tubule, it is released into the lumen of the seminiferous tubule, and then it travels towards, basically out of the lobule through these tubules right here, which for some reason are not labeled on this diagram. But the ones that are marked in orange, these are the straight tubules. Their name kind of makes sense. They're straight, right? So they're very short and just straight. They're not as convoluted as we see the seminiferous tubules. Straight tubules then lead the sperm into this kind of a network of these tubules that you see here. See how they're kind of interconnected and all that. That is the reed testes. Reed testes is in the mediastinum testes. And yeah, so it's just kind of an anatomical region holding this reed testes here. From the reed testes, sperm exit through the efferent ductules or efferent ducts. Sometimes they just are called efferent ducts. And then those lead the sperm into the tubules of the epididymis. And epididymis is this thing, okay? That's your epididymis. And the epididymis, just the epididymis tubule, if you were to unwind the tubule of the epididymis, it is anywhere between 4 to 5 meters long. So that's a very long tubule. And I should have grabbed something that's a meter. Usually in the classroom, I have a meter stick. I do not have a meter stick in the house. But 1 meter is about 3 feet, okay? So 4 to 5 meters, multiply that by 3, and that's the length of the tubules in just the epididymis in feet. So that's the epididymis. Epididymis is a site of maturation for sperm. So this is where the sperm complete their maturation. This is where they gain their ability to move. And we know that this is where they learn to move, because if you were to extract sperm from the testes before they enter epididymis, the sperm do not move. Once you get them after they pass epididymis, they do move. And about the movement of the sperm do not actually swim in the tubules. The way that sperm are moved through the tubules inside of the male's body is by peristaltic contractions within these tubules. Okay, so there are different ways that we are moving sperm through the tubules. Mostly it's going to be those peristaltic contractions. but the sperm will not swim inside of the male's body. It takes a lot of energy for the sperm to swim, so it's not going to waste it in the male's body when there are other mechanisms that can move the sperm through. Sperm are saving all their energy to swim until they're out of the body, so that they can ultimately, hopefully, get to the oocyte. That's their ultimate goal in life, is to find an oocyte and fertilize it. So that's the epididymis. From the epididymis, the sperm then enter the vas deferens. And for the vas deferens, I'm going to go back to this image right here. Okay. So after the epididymis, the sperm go into vas deferens. Okay. The vas deferens goes through the spermatic cord. It passes on the side. Okay, this is not really touching the pubic symphysis here because spermatic cord is kind of in the inguinal canal, right? But kind of height-wise, that's where it is right here. Then the vas deferens runs along the sides of the urinary bladder until it reaches where the ureters connect to the urinary bladder. The vas deferens loops above the ureter, kind of behind it, and then turns downwards on the posterior side of the bladder. This is where that vas deferens widens, and it's not super obvious that it's a little wider, but it is a little wider. We'll see it really well when we have a posterior view of the penis. Okay, after the sperm passes through the vas deferens, the vas deferens, the ampulla of the vas deferens is going to fuse with the seminal vesicle opening. Okay, so seminal vesicle has an exit or a tubule, and then the vas deferens has a tubule, and these two will then fuse together and create what we call ejaculatory duct. That's this right here. Okay, so that's your ejaculatory duct. It runs through the prostate gland and it connects to the prostatic urethra. Okay, this in the blue is the prostatic urethra. Okay, and then in the yellow is ejaculatory duct. After ejaculatory duct merges with the prostatic urethra, that little short part, that's still prostatic urethra. So we don't change the name. Like if you remember when we talked in the digestive system, we talked about all the ducts, and every time something connected to something else, we changed the name. It's not like that here. Okay, you have ejaculatory duct, it just empties into the prostatic urethra, and then that's your prostatic urethra. From the ejaculatory duct, then we go to the rest of the prostatic urethra. Then the sperm would go through membranous urethra, which I'm gonna do in orange. Orange was not a good choice, but it's there. Maybe let's do purple. It's not really super bright either, but it's the shortest part of the urethra, of the male urethra, and it runs through the urogenital diaphragm. And we already know this from the urinary system. And then the longest part of the urethra in males is the spongy urethra, and that's this. The spongy urethra leads ultimately the sperm out of the body, yeah, that's that. That's the pathway of the sperm through the male's body, starting with the seminiferous tubules all the way to the spongy urethra, and then the external urethral orifice is the opening right here. Okay, so to summarize that again, we start in the seminiferous tubules, then straight tubules, reed testes, then epididymis, then vas deferens, then ampulla of the vas deferens, then ejaculatory duct, prostatic urethra, membranous urethra, spongy urethra. Okay, make sure that you are able to tell me this pathway. Okay. You will probably be putting these in order of how sperm travels through the male's body. That's, I will definitely ask you about the pathway of the sperm. So can't guarantee you that it's going to be order. It might be mad. I don't know what kind of question it will be, but I will ask you about this. So know it. Okay. So let's look at how the sperm is formed then. So now we know how the sperm travels through the male body. But let's see how the sperm is actually formed. So in females, we looked at the process known as oogenesis, and that one was a little more complicated because we ultimately ended up with just a single cell that had a potential to become the ovum. But in males, it's basically the same as what you see on meiosis, okay? That's pretty much what is happening there. So I'm gonna bring this picture back here because remember, spermatogenesis or formation of these sperm cells happens within the seminiferous tubules. The spermatogenesis begins with a cell known as the spermatogonium. And that's kind of similar. Terminology is kind of similar with the female, except female, the beginning of the word had oo. In the males, the beginning of the word has spermato. Okay, and then the other things are the same. So we begin with a cell known as the spermatogonium. Spermatogonium is a diploid cell. It has 23 pairs of chromosomes, and it divides by mitosis. Okay, so up to this point, it's similar as far as what happens with the females. We had the oogonium. It also divided by process of mitosis, right? It's a diploid cell that divides by process of mitosis. Except the difference between males and females is in females, this process began before females were even born. In males, spermatogenesis does no part of spermatogenesis begins before puberty begins, okay? So that's a difference there. So when the spermatogonium divides by mitosis, it produces two cells. One of these cells will become primary spermatocyte. The other cell will become a new spermatogonium. Okay, again, I want you to think back to oogenesis. When the oogonium divided, we got two primary oocytes. We didn't create a new oogonium. So once you divide the oogonium, you're done. It's gone. You just have your primary oocytes. But with the spermatogenesis, every time a spermatogonium divides, it creates a new spermatogonium. This gives males the ability to produce sperm forever, okay, until they die. And that's males generally produce gametes until they die, okay, unless they have some condition, but normally that's what happens, okay, because they constantly have a new supply of spermatogonium, okay. Once females divide our oogonia, they're gone, okay. That's why females have a biological clock, okay. You have a limited amount of oocytes that your body has and will produce. But with the sperm, you have unlimited number of sperm because every time you divide that spermatogonium, it creates a new spermatogonium. Okay, so that the primary spermatocyte then, that is the cell that's going to enter meiosis. So think in here, remember this. This would be in the spermatogenesis, this would be that primary spermatocyte, okay? So that's our primary spermatocyte. So the primary spermatocyte then will divide, it will go through the meiosis 1 process. It will divide into two secondary spermatocytes. And then each of those secondary spermatocytes are going to go through meiosis 2. And then the meiosis 2 will then result in 4 non-identical daughter cells, which are known as the spermatids. And spermatids are not sperm yet, okay? They are actually still round. They're much smaller than the original cell. So you can see that the amount of cytoplasm decreases as we're getting towards the final product, towards the sperm. So spermatids are still round. They don't have that typical shape of the sperm. And spermatids then will go through the process of spermiogenesis. And spermiogenesis is just the final modifications of the spermatids to give the sperm the actual shape. And so you can see that here, that's shown right here. You can see that the cytoplasm is rearranging, it's getting smaller, The nucleus becomes the major component of the head of the sperm. And then the mitochondria that are in the cytoplasm, because these spermatids are going to have a lot of mitochondria. The mitochondria will then migrate into this area right here, and that is the forming midpiece. Okay, so the final appearance of the sperm is you have three major parts. You have the head, midpiece, and tail. Head contains the nucleus, the genetic information, but not really much cytoplasm, as you can see. Midpiece is going to have high concentration of mitochondria. Again, not too much cytoplasm. Cytoplasm is extra weight. We don't need it, okay? So you're trying to get rid of as much weight as possible here. And then the flagellum or the tail is the last part of the sperm. At the top of the head of the sperm is a structure that we know as the acrosome cap. And acrosome cap contains enzymes that will help the sperm penetrate the zona pellucida, as well as push the cells of the corona radiata apart should the sperm encounter the oocyte. Okay, if the sperm meets oocyte, it needs to be able to get through the zona pellucida and the corona radiata. Okay, so here we see that final appearance of the sperm here. Okay, so mitochondria in that midpiece, this serves like a battery pack for the cell, for the sperm. Okay, so that's what makes it go. Okay, so that's spermatogenesis here. All right, what else? So we talked about all the different ducts and how the sperm move. Now let's talk a little bit about the accessory glands. And we can see some of them here. So the three major accessory glands that are important are the prostate gland, seminal vesicles, and the bulbourethral gland. Bulbourethral gland this is a pair of glands here, and those are these smaller ones right here. They secrete mucin that is going to later form mucus, and this protects urethra, and it's also a lubricant for the male reproductive system. The prostate gland secretes various secretions, which include citric acid. Citric acid is a nutrient for sperm, actually. There is also prostate-specific antigen that comes from the prostate gland, and this is an enzyme that helps liquefy semen. And then also seminal plasmin. This seminal plasmin fights the urinary tract infections in males. So another reason why males are less likely to get urinary tract infection is that they have this seminal plasmin. They have this protection from UTIs, and females don't have this. And then our seminal vesicles, and prostate gland is this gland right here, okay? And here on this view, we can also see how the seminal vesicle, which is this, merges with that vas deferens. And here you can see that the ampulla of the vas deferens really is a little wider. And then here you see the two ejaculatory ducts right there, okay? So seminal vesicles are also, there's a pair of them. And seminal vesicles produce alkaline fluid containing fructose, and this serves for energy for the sperm. And also seminal vesicles release prostaglandins. Prostaglandins help dilate external os of the cervix, and this makes it easier for the sperm to enter the woman's body. So that's what that does. And yeah, already talked about bulbourethral gland. So what else on here? The next section or the next topic would be talking a little bit about the semen, because we mentioned semen. The semen is the word that we use for seminal fluid plus sperm. So when the sperm mixes with all the secretions from these accessory glands, it is termed semen. when it is released from the body, it is termed ejaculate. The volume of the ejaculate is about 3 to 5 milliliters. It's not a very large volume. And this amount of ejaculate contains normally between 200 to 500 million sperm. It takes about 2 weeks for the sperm to get through from the seminal tubule or seminiferous tubules into the ejaculate. And so it's what's interesting is that males that have frequent ejaculations actually tend to have lower sperm count because their body is not able to catch up with the excessive release of the sperm. Okay, volume doesn't change though. Sperm are the smallest cells in the body. Oocytes are the largest. Sperm are the smallest. So even if the sperm count is lower, volume generally really remains the same. So that's that. Okay, another accessory structure is the penis here. Make sure that you're able to label the parts here. I think this is external, well, and internal as well, but make sure that you're able to label the root, the body, the glands, the orifice, and everything that's labeled on here. Okay, again, that's just the labeling practice. On here, we can see the different erectile bodies. There are 3 erectile bodies that compose the structure of the penis, and these are corpus spongiosum and corpora cavernosa, 2 corpora cavernosa. Corpora is a plural of corpus, so corpus is 1, corpora are 2, and corpus spongiosum is the part of the penis that the spongy urethra runs through, and that is why spongy urethra is called spongy, because it goes through that corpus spongiosum. And then corpora cavernosa are the 2 other erectile bodies. These have a larger, larger spaces in them. And so this is a cross section here of the penis. And you can see those 3 erectile bodies. Okay, they're kind of cylindrical structures. And so here are this is 1, this is 2, that's the 2 corpora cavernosa, or 1 corpus cavernosum, if we're just pointing to 1. And then we have this 1 corpus spongiosum. Okay, so when you look at this, it kind of looks like a bug face, right? The 2 big eyes and the mouth. See, once you see it, then you can't unsee it. So anyway, so these erectile bodies have to do with how the penis functions. And there is it labeled on here? There is actually a layer of muscle here that surrounds the erectile bodies, but that's not what causes an erection. Okay, the thin layer of muscle that's actually present in the penis has the opposite effect. It actually causes erection to be over. So that muscle layer is involved after orgasm. So what happens during this really leads us to the male sexual response here, because it explains why the erectile bodies are like this. So there are, as you can see, this looks kind of spongy. Even the corpora cavernosa looks spongy. All these spaces here are spaces. And these spaces are going to be filled with blood. So there are, the first phase of the male sexual response is the excitement phase. And during the excitement phase, the blood flow is redirected. It's directed towards these erectile bodies. And the blood starts filling all these spaces in these erectile bodies. As the blood fills these spaces, this causes the erectile body to get filled with blood. And as it fills with blood, it becomes more and more rigid. Well, as it becomes more and more rigid, it constricts the veins that normally take the blood away from the area. And so the more blood that comes in, the more the veins are constricted and the blood cannot leave. Essentially, the blood is getting trapped in these erectile bodies during the excitement phase. there is a parasympathetic innervation that releases a vasodilator, and this is going to help with bringing the blood in. So it was, as you vasodilate the vessels, more blood can come in, okay, faster as well. Okay, so that's the excitement phase in males. It's basically just filling these spaces with blood. Then is the orgasm. During orgasm in males, the vas deferens is going to move the sperm towards the urethra. So the smooth muscle within the vas deferens will start contracting, and it pushes all the sperm towards the urethra. Urethra also has a smooth muscle layer, and that one is also going to start contracting, and that helps with ejecting the sperm out. Accessory glands are also going to release their secretions during orgasm, so the sperm mixes with all these secretions as it leaves the body. The internal urethral sphincter contracts during orgasm, and this is important because you want to make sure that the sperm is going out of your body and not up the urethra into your bladder. And so we see the contraction of the internal urethral sphincter for that reason. So it is nearly impossible for males to urinate even with an erection, okay? And that's because that internal urethral sphincter is already contracted. Then the ejaculation is controlled by sympathetic innervation. The rhythmic contractions of the smooth muscle of the urethra are controlled by the sympathetic innervation. So both parasympathetic and sympathetic innervation aid with the process, with the male sexual response here. So that's that. After orgasm, males then go through resolution phase. And resolution phase is a phase of relaxation. This is when the smooth muscle is going to, in the urethra and the vas deferens, this smooth muscle is going to relax, so it's no longer contracting during resolution phase, but the smooth muscle that surrounds the erectile bodies will now contract, and it will help with expelling the excess blood out of those erectile bodies so that the erection is reversed, I guess. There's a part of the resolution phase that is known as the refractory period. So refractory period and resolution phase begin at the same time, right after orgasm is finished. And the refractory period is the period during which it is impossible for males to have another erection. So it is part of resolution phase. Resolution phase would maybe be longer, or maybe a refractory period will be longer. I don't know, this is subjective. And a refractory period is different in every male. There's one trend that's kind of common between all males, and that is that a refractory period generally increases with age. So it gets longer and longer as males age. But it's different for every male as far as the length of the actual refractory period. So that's male sexual response. I said that I would leave the female sexual response for later until we are also talking about the male sexual response so that we can compare the two. So for female sexual response, the first phase is also excitement phase, and it also involves redirecting of blood towards the reproductive structures. The blood is going to rush towards the vagina as well as the uterus, and the uterus actually becomes erect during the excitement phase. So you know how normally it leans on the bladder. During the excitement phase, it kind of stands up a little bit. The glands within the vaginal walls begin to produce mucus. This is necessary for lubrication. We are seeing heart rate, blood pressure, and respiratory rate increase. This is also happening in males, the heart rate, blood pressure, and respiratory rate. And so that's excitement phase. If the excitement phase is followed by orgasm, orgasm involves intense feelings of pleasure accompanied by the rhythmical contractions of uterus and vagina. So the orgasm involves contractions of smooth muscle as well here. In males, it was the vas deferens and the urethra to get the sperm out. In females, this is contractions of the uterus and the vagina. Then after the orgasm would be the resolution phase. A resolution phase means return to pre-excitement state. However, females do not have a refractory period as part of their resolution phase. Females are capable of multiple orgasms in a row, so they do not require a refractory period like males. And that is the sexual response for both males and females. Now we're going to look at the last part of the chapter here, and that is development of the internal and external reproductive structures. So if we look at this image right here, I don't know what that was. I don't know if you heard that, but there was some strange noise coming from outside. Sorry about that. Okay, so we're gonna look at development of the internal reproductive structures here first. This image shows us, and I already showed you this when we talked about the homologous structures, right? So this image shows you the picture of an embryo between 5 to 6 weeks. And if you wanna, kind of forgot to give you a page reference here, but if you wanted to see where we are in the textbook, this is that section 28.5. This is page 1140. So a sexually indifferent stage, you can't tell the difference between a male and female just by looking at the reproductive structures. They look the same. At this point, both mesonephric and paramesonephric ducts are present. The mesonephric ducts are also known as the Wolffian ducts, and these are the male ducts. And then paramesonephric ducts are also known as the Mullerian ducts, and these are going to form the female duct, okay? Usually only one of these is going to remain, but initially both of them are present. So what happens and how does the body know which one to keep? Well, it's all about your genetics. So it depends on what sex chromosomes you have. If you have a Y chromosome, if you're a male and you have an X and Y chromosome, that Y chromosome carries this region on it, which is known as the SRY region. The SRY region stimulates production of androgens, and these androgens stimulate the male development. So if you have Y chromosome, it will stimulate formation or development of the mesonephric duct. Okay, that's, well, it's that green stuff. Well, really the purple stuff. Okay, so this will be stimulated to continue maturing. As the male duct, the mesonephric duct matures, this is going to start forming anti-Mullerian hormone. Okay, anti-Mullerian hormone, we said that paramesonephric duct is also known as the Mullerian duct, right? And so the anti-Mullerian hormone is designed to start degrading the paramesonephric duct or the Mullerian duct. So if you have the Y chromosome will stimulate male development, and as the male structures are developing, they're releasing chemicals or signals that stimulate degradation of the female duct system. So female won't develop, okay? Now, if you are an X and X, so you're a female, and you do not have the Y chromosome, then there is nothing that would stimulate the development of the mesonephric duct. Therefore, mesonephric duct is not supported, it's not promoted to develop, and it's not releasing the anti-Mullerian hormone. And so if you don't have Y chromosome, the paramesonephric duct is allowed to develop, while the mesonephric duct is not stimulated to develop. Okay, so that's the difference here. You can see that right here. So you have this is the male, right? Oops, male. All right, and then female on the other side, and yeah, so you can see how about weeks 10 to 12, the female duct begins to fall apart and degrades, and then you don't really have it present at all at birth. Okay, now this is what generally happens. Okay, so that's the internal reproductive structures. Now let's look at the development of the external genitalia, and initially there is this sexually indifferent stage also, even for the external genitalia. So looking at a week 6 embryo here, you can see that there is both males and females look pretty much the same. Okay, and then as we get a little later on into the pregnancy of week 12, this is where they still look pretty similar. Okay, so you see that in a female, this part of that that genital tubercle that we saw on the previous picture is the developing clitoris, and in males that part will develop into the glans of penis. This is another example how these structures are homologous. They are originating from the same tissue. They have pretty much the same structure function, right? So both clitoris and glans of penis contain the tissue that reacts to stimulation. So that's that. Then labia minora and body of penis also originated from the same tissue, as well as labia majora. And then this would be the scrotum, right? Developing scrotum. So you can see that initially they look the same, but then depending on the presence of that Y chromosome or absence, if it's not there, they develop into different form, okay, different structure. To finish off this section, the very last page, the 28.5E, is a section on puberty and menopause. So just be aware of what puberty is. Puberty is the time when the hypothalamus begins releasing that gonadotropin releasing hormone, and then that stimulates the anterior pituitary to release the luteinizing and follicle stimulating hormone. This is in both males and females. And then those cause a release of the sex hormones. When it begins, that puberty isn't easy reading. So I have a 10-year-old now, which, yeah, she's 10 and a half. Definitely can tell that there are hormones already. So Lord help me. But anyway, so that's that about puberty. The girls do begin puberty earlier than boys generally. And for girls, like sometimes people think that for girls, in order for them to start puberty, they have to begin their menstruation. This is not the truth. Puberty begins even a couple of years before menstruation might actually happen in a girl. So there are changes noticeable before menstruation begins in girls. But boys are generally about a year or two behind girls as far as puberty beginning. As reproductive system and aging, for females, when females age, they actually eventually stop maturing gametes. And so females don't produce gametes forever, okay? Around later 50s or not later 50s, later 40s or early 50s is when females enter menopause. And just like puberty is not like a boom, like it starts and, it's kind of a gradual process. Menopause is a kind of a gradual process as well. So eventually women stop producing gametes. The stop of gamete production is associated with the reduction of the hormones that the woman produces or releases. And so this reduction of hormones is, actually, I'm gonna stop the sharing here. So the reduction of hormones has some fun side effects. So for women, you know, not only do we have to go through the periods throughout our adulthood, since puberty until adulthood, then we have this awesome menopause. So this reduction of hormones has side effects such as hot flashes, reduction of hair on head, but increase of facial hair and just random hair. And then women become more prone to osteoporosis and heart disease. And this is because the reduction of hormones affects our ability to absorb calcium and process calcium. And so osteoporosis is often diagnosed in women that are going through the menopause. So my kind of a story from my life, my mom had to have she had to have hysterectomy when she was, I think, 45, but she wasn't starting menopause yet. And so hysterectomy, they did a complete hysterectomy where they removed the uterus as well as the ovaries. So everything was taken out. And within a year, she was diagnosed with osteoporosis. So it definitely messed up her calcium balance there. Fortunately, not heart disease. She also did have a lot of troubles with hot flashes and such. So throws off the whole body. So a hysterectomy is like an instant menopause. You know, regular menopause, at least it kind of gradually happens, although all these usually do happen. But anyway, so that's that. For the males, males do not go through menopause. Males, we call this aging of male reproductive system male climacterics. Males might experience slight reduction of the interstitial cells or those Leydig cells that produce testosterone. So they might experience slight reduction of testosterone, but generally it's not something that is significant. Sometimes there are some symptoms for males with this that are associated with the reduction of testosterone, which include mood swings, decreased sex drive, as well as hot flashes. So yeah, sometimes when my husband is grumpy, I tell him he's turning into grumpy old man, which I guess reduction of testosterone would kind of go with that stereotype. Not saying that all old men are grumpy. They're not. Okay. It's just that stereotype thing. But males do produce gametes pretty much for the rest of their life. Okay. One other thing that happens as males age, and this is normal, is that the prostate gland enlarges. However, prostate cancer is a fairly common cancer, and so males should go get their prostate checked every year, I believe. I think they started after 50 or maybe after 40. I don't remember. I know mammograms are 40. But some doctors don't do prostate checks on younger people as part of their regular physicals. But males, make sure that you go have your physicals. Same for ladies. You know, women, we have to go get pap smears all the time, and then mammograms I hear are not all that fun at all either. But you know, we do that to screen for cancers, and prostate cancer is fairly treatable if it's caught early. And so it's important that you have those checks for both ladies, your mammograms, your pap smears, and for males, go get your prostate checks. Okay. So I think that's it for this chapter, and I'll see you in the next session, and that will be on the human development, which we will not we're not going to do human development in too much of depth, but we will talk about some development, and then we'll talk about pregnancy, and then last part will be heredity. So we'll be doing some Punnett squares and fun stuff. All right, so I'll see you later.